Hello and a very good day to all the assessors and viewers. I am Joshua Lojuni, a student in the Master of Science in Molecular Medicine program, and today I'll be presenting on my research proposal titled Cytotoxic and Apoptogenic Effects of Syzygium aqueum extracts on pancreatic cancer cell line. So, the contents that will be covered in this presentation includes the introduction, literature review, hypothesis, research question and objectives, methodology, expected outcomes, timeline, budget, and references. Let's start off with the introduction. Does nature hold the key to safer cancer treatments? Cancer is defined as the uncontrolled proliferation and spread of abnormal cells affecting healthy tissues and organs. After the COVID-19 pandemic, cancer remains the second leading cause of mortality due to the delay in diagnosis and treatments. It is projected that there will be over 2 million new cancer cases and 600,000 cancer-related deaths by 2025. There are around 277 different types of cancers. Pancreatic cancer, abbreviated as PC, is the third leading cause of cancer-related deaths. It is projected to become the second leading cause by 2040, surpassing colorectal cancer. The most common type and most lethal type of PC is the pancreatic ductal adenocarcinoma. The current treatments for PDAC are often ineffective, prone to resistance, and cause significant side effects. Now, on to nature's potential solution. Syzygium aqueum, commonly known as watery rose apples, is a plant belonging to the Syzygium genus and the Myrtaceae family. It grows in tropical and subtropical regions and is rich in phytochemicals that contributes to its medicinal properties like anti-cancer. Recent studies have shown that it can selectively kill various types of cancer cells by inducing apoptosis. Since dysregulation of apoptosis is a hallmark of pancreatic cancer, Syzygium aqueum could be a potential alternative or complementary treatment. However, this presents a research gap, as studies on its anti-cancer mechanisms, optimal dosage, and selectivity are often limited. Moreover, there are no studies conducted on PC models yet. Hence, the aim of this study is to evaluate the cytotoxic and apoptogenic effects of Syzygium aqueum on pancreatic cancer cell line. Now, on to the literature review. PC can be categorized into adenocarcinoma or endocrine tumors based on the cellular origin. PDAC is an adenocarcinoma subset and the most lethal type, with a 5-year survival rate of only 12%. Since it's also the most common, accounting for 80 to 90% of cases, PDAC is the primary focus in PC research. Its lethality is due to its aggressive and asymptomatic progression and strong resistance to treatment. PDAC develops from non-invasive precancerous lesions in the pancreatic duct. Its dense desmoplastic stroma complicates molecular analysis where 90% of PDAC cells are non-neoplastic. Additionally, molecular variants exist, but somatic mutations are the main causes contributing to by factors like aging and smoking. As shown in Table 1, key oncogene mutations includes the KRAS gene, and key tumor suppressor genes involved are CDK2NA, TP53, and SMAD4. Oncogene mutations drive uncontrolled proliferation, while tumor suppressor genes hinder its regulatory mechanisms and allow cells to evade cell death. Additionally, about 10% of PDAC cases are actually hereditary, associated with syndromes like the Peutz-Jeghers syndrome. These individuals acquire tumor suppressors STK11 gene mutations, leading to the increased risk of acquiring PDAC. Now, let's review on the current therapies for pancreatic cancer. Surgical resection remains the best option, but only 15 to 20% of patients are eligible due to the distant metastasis and tumor being too close to critical vasculatures. Chemotherapy regimens like FOLFIRINOX are used to shrink tumors and facilitate downstaging, but also have significant side effects and often lead to chemo resistance. Radiotherapy, on the other hand, kills cancer cells with X-ray, but also damages nearby healthy tissues. Furthermore, tumor hypoxia promotes further resistance. Targeted therapy was introduced to overcome the above challenges by targeting specific molecular pathways. However, all PC-targeted treatments have failed in clinical trials, except for pembrolizumab, which is only effective for specific genetic mutations and is generally ineffective in PDAC due to its low immunogenicity environment. As observed in this illustration, the main factor that contributes to the development of treatment resistance includes PC's microenvironment, which facilitate in evasion of apoptosis, immunosuppression, a dense tumor stroma which acts as a barrier to prevent drug penetration and proper vascularization, and drug efflux mechanisms. Now, moving on to Syzygium aqueum. Syzygium aqueum has been traditionally used in folk medicine using leaves, barks, and fruits. It contains active compounds such as flavonoids, terpenoids, and phenols. These compounds are determined to contribute to Syzygium aqueum cytotoxic effects by inducing apoptosis with the addition of inhibiting proliferation, angiogenesis, and metastasis. Studies have shown that Syzygium aqueum extracts have significant cytotoxic effects on breast cancer cells, especially the MCF7 or hormone-dependent breast cancer cell line, where polyphenols present were suggested to disrupt estrogen signaling and ultimately cell proliferation. Additionally, a compound called chalone, isolated from S. aqueum, showed that its primary mechanism was apoptosis induction through immunoblot assays. Importantly, Syzygium aqueum showed no cytotoxicity against normal fibroblast cells. Moreover, other notable studies on Syzygium aqueum cytotoxic effects on breast, lung, and cervical cancer cells are summarized here in Table 2, demonstrating varying levels of toxicity based on their IC50 values. However, no studies have yet to be tested its effects on pancreatic cancer cells. In summary, Syzygium aqueum has promising anti-cancer potential, but requires novel and further study in pancreatic cancer models. Moving on to the hypothesis, the null hypothesis is that Syzygium aqueum extracts has no cytotoxic or apoptogenic effects on pancreatic cancer cells, whereas the alternative hypothesis is that Syzygium aqueum has cytotoxic and apoptogenic effects on pancreatic cancer cells. The research question is that do Syzygium aqueum extracts have cytotoxic and apoptogenic effects on pancreatic cancer cells? And the objective of this study is to determine the cytotoxic and apoptogenic effects of Syzygium aqueum extracts on pancreatic cancer cells. Moving on to the methodology section, this research will undergo plant extraction, cell culture preparation, MTT assay, doubling time assay, Koch staining, and statistical analysis. Firstly, in the plant extract preparation methods, 100 grams of dried Syzygium aqueum leaves and stem barks will be obtained from a farm in Perak. 4 solvents will be used for the extraction process, producing 8 extracts in total, 4 from the leaves and 4 from the stem barks. This enables the isolation of a wide range of phytochemicals with different properties like polarity, determining which extract works best. The extraction process involves incubation with the respective solvent for 72 hours in the dark, filtration, solvent evaporation, and drying. Next, in the cell culture preparation step, the pancreatic cancer cell line BXPC3 and the normal fibroblast cell lines 3T3 will be cultured, mimicking the PDAC and healthy cells respectively. Cells will be grown in the DMEM medium supplemented with fetal bovine serum, penicillin, and streptomycin at 37 degrees Celsius with 5% carbon dioxide. Now, on to the MTT assay, where this assay measures cell viability. The principle is based on viable cells with functional mitochondria and active dehydrogenase, converting MTT into formazan crystals. Cells will be seeded and incubated 24 hours at 37 degrees Celsius, treated with different concentrations of extract, which includes 0, which is the controls, 5, 12.5, 25, 50, 100, 150, and 200 micrograms per milliliter concentrations, and will be re-incubated at 37 degrees Celsius for 24, 48, and 72 hours to evaluate time-dependent cytotoxicity. Next, MTT assay will be added to and incubated further. DMSO will then be added to dissolve the crystals, and the absorbance will be spectrophotometrically measured, and triplicates will be performed for this experiment. As for the calculation, the percentage of cell viability will be calculated using the absorbance of treated cells over the absorbance of control cells, where IC50 values will then be determined via graph interpolation. Subsequently, selective index will be calculated by comparing cancer and normal cells IC50, where an SI of 10 or above indicates excellent selectivity. Next includes the cell doubling time assay, which compare how the extract affects the proliferation rate of cancer cells versus normal cells. Cells will be treated with IC25, IC50, and IC75 concentrations, and after treatment, cells will be stained with trypan blue and live cells will be counted. The doubling time will be calculated by dividing the duration by the number of generations. Similarly, triplicates will be performed. Moving on to the Horst staining, which validates the apoptogenic effects of Syzygium aqueum, cells will be treated with the IC25, IC50, and IC75 concentrations and incubated for 72 hours. Cells will then be stained with Horst 33258 and fluorescence microscopy will be used to observe the nuclear changes. The percentage of apoptogenic cells will be calculated using the formula of the number of apoptogenic cells over the total cells multiplied by 100. Similarly, triplicates will be performed. Finally, statistical analysis will be performed. All data will be expressed as mean plus minus the standard deviation due to the triplicates performed. SPSS version 16.0 will be used, where one-way ANOVA, which compares the variance of the group means, followed by Tukey's post-hoc test, which conducts the pairwise comparisons between group means, will be applied. A p-value of less than 0.05 will be considered statistically significant. As for the potential implications, Syzygium aqueum could offer a safer, natural, and more targeted treatment for pancreatic cancer. This study could contribute to future pharmacological research, such as providing additional insights into Syzygium aqueum cytotoxic mechanisms and applications. Moreover, if this research is successful, preclinical trials using animal models to determine its pharmacokinetics, efficacy, and toxicity can be potentially be expedited. Now, on to the expected outcomes. In the MTT assay, Syzygium aqueum is expected to show selective cytotoxicity against pancreatic cancer cells. This is reflected by the treated PC cells acquiring lower absorbance values, indicating fewer viable cells. It is also expected that an increased extract concentrations will enhance cytotoxicity, and the IC50 value for cancer cells should be lower than the normal cells. In the doubling time assay, treated PC cells should show increased doubling time, meaning reduced proliferation rate, whereas untreated cells will show minimal to no changes. In Horst staining, treated PC cells will show bright, condensed, and fragmented nuclei indicating apoptosis, whereas untreated cells would show dim, intact, round nuclei indicating no apoptosis. Penultimately, let's brief through the project's timeline. From March to June 2025, literature review and proposal writing will be prepared. July to August 2025, we will obtain the approval from the IMU-JC board. September to October 2025, cell and extract preparation and MTT assay will be performed. November 2025, doubling time determination and Horst staining will be carried out. December 2025 to January 2026, statistical analysis will be performed. And finally, October 2025 to January 2026, dissertation writing will commence. The target completion date for this research is in January 2026. Finally, let's brief through on the budget. A total of 10,000 ringgit Malaysia will be used for this research project, where 30% will be used for cell culture consumables and 70% will be used for experimental reagents and kits. And with that, this concludes the presentation. Here is the list of references. And lastly, I would like to thank you all for your attention, and I'm happy to take any questions.